Hello, wonderful people. It's Medicosis Perfectionalis, where medicine makes perfect sense. Welcome back to my biochemistry playlist. In previous videos, we talked about carbohydrate metabolism, glycolysis, and gluconeogenesis. We talked about protein metabolism, proteogenesis, and proteolysis. And we also talked about lipid metabolism, lipogenesis, and lipolysis. And we also talked about the lipoproteins, the LDL, VLDL, HDL, IDL, etc. Today we shall expand on the topic of lipogenesis, genesis of fat, storage of fat. One of the ways your body stores fat for future use is by converting tiny pieces of fat into bigger pieces of fat. The big pieces of fat are called triglycerides. Did you know that your body has the capacity of converting carbs to fat? And that's why if you eat a lot of sweets, tons of desserts, you will literally become fat. Triglyceride synthesis is an anabolic pathway where you are building something up instead of tearing something down. Click the like button, click the subscribe button, and let's get started. There are more than 200 biochemistry videos in my biochemistry playlist. There is one for fatty acid synthesis and another for fatty acid degradation. There is a video on cholesterol metabolism and the lipoproteins, another on signaling lipids, terpenes and trypanoids, and steroids, another on phospholipids, sphingolipids, cholesterol, and wax, another on insulation and saponification, another giving you a broad overview called lipid metabolism. Even the diseases are covered. There is a video on fatty acid oxidation disorders like the carnitine deficiencies, another on hyperlipidemias, especially the familial ones, all of the 5 types are discussed, and another on sphingolipidosis. Who needs a textbook when you can just watch Medicosis? You can find all of these videos in my biochemistry playlist. Why do you need lipids in your body? For many reasons. Number 1, they provide structure, such as the structure of your cell membrane. You have trillions of cells in your body. Each one has a cell membrane, and the cell membrane has lipids, especially phospholipid and some cholesterol. Second, for signaling, as in the second messenger systems. And I've talked about these signaling pathways before. You can find these videos in my biochemistry playlist and in my physiology playlist. Third, lipids are important for storage in order for your body to store energy for a rainy day. That will come later. But why does the body prefer to store energy in the form of lipid? Why not protein? Why not carbohydrate? And the answer is because lipid is dense. Lipids will provide you with more calories per unit stored, more calories per gram, as we will discuss soon. When you eat, you're either eating carbs, proteins, or fat. The big plant carb is called starch. The big animal carb is called glycogen. You start with polysaccharides and then you digest them into disaccharides and monosaccharides. The monosaccharides are glucose, fructose, and galactose. You can absorb those from gut to blood. Next, when you eat proteins, they are big. They are polypeptides. Break them down. Oligopeptides. Break them down. Dipeptides. Break them down. Monopeptides. Amino acids. When you eat down fat, you're eating big fat, such as triglycerides or others. Then when you break them down, they become glycerol and free fatty acids. Don't ever forget that. When you break down triglycerides, they become glycerol and free fatty acids. Ergo, we can say that conversely, if you want to build up some triglycerides, what are the raw materials that you need? You need glycerol and free fatty acids. Make no mistake about it. You can obtain both the glycerol and the fatty acids from glucose. And that's why when you eat lots of sugar, glucose, you will literally become fat because you will store tons of triglycerides because glucose will give you this and will give you this. And when you combine this with this, you're gonna get this. All of that big fat will get stored, and that's why your belly is getting so big. Next, did you know that when you digest fat, you will not require 1 molecule of water, but 3? That's weird. That's not the case with carbs or proteins, but fats are special because you'll need 3 molecules of water, and that's why when you eat a heavy, fatty meal, you're gonna feel thirsty. Why is this? Because your body is using all of the water that it can find to digest all of that fat. What types of fats are you eating? Could be triglycerides, cholesterol, cholesterol esters, phospholipids, even fat-soluble vitamins. Break down the triglycerides into 3 fatty acids and 1 glycerol. Not glycerol per se, but 2 monoacyl glycerol. Let me tell you something. When you hear the word acyl, think fat. This is going to help you a lot in biochemistry. When you see acyl, think fat. But acyl is not the same as acetyl. Acetyl is the centerpiece. It's the hub. Acetyl can be part of carbs, can be part of fat, it can be part of protein. But when you hear acyl in biochemistry, think fat. But what is acyl? Well, acyl is R bonded to C, double bonded to oxygen. Alkyl group, carbon, oxygen. If you have watched my biochemistry videos on metabolism, you will remember my 10 commandments of metabolism. Number 1, thou shalt understand the difference between the insulin world and the glucagon world. Number 2, glucose is key. It's your brain's favorite source of energy. Glucose can also help you make fat. And what's the name of the hub or the hinge or the sun around which all of the biochemical pathways revolve? It's acetyl-CoA. Commandment number 1, thou shalt understand the difference between the insulin world and the glucagon world. Insulin land is the feeding state. Glucagon land is the fasting state. Think about it. When you are feeding, you want to build up something. I eat so that I can build up proteins in my muscles. I eat so that I can build up glycogen in my liver. I eat so that I can build up fat in my belly. I need to be more respectful. I should say I will build up fat in the subcutaneous tissue, such as the superficial fascia, otherwise known as hypodermis. Please note that the human scrotum does not have superficial fascia. And that's why if you examine a morbidly obese male patient, the scrotum never gets fat. The same holds true for the male organ of copulation. These organs either lack the superficial fascia or they have a superficial fascia that's devoid of fat. Okay, what if I am not in the feeding state? What if I'm in the fasting state? Oh, that's a glucagon world. It's not just glucagon, it's glucagon and his friends. Who are glucagon's friends? Introducing epinephrine, introducing cortisol, and sometimes thyroid hormone or thyroxine comes to play. Hey glucagon, what are you going to do? Well, I'm in the fasting state, so I need energy. The body needs energy because we're not eating. So let me break down the sources of energy that have been storing from before, and let me break them down into readily available sources of energy because I need ATP right now. So the proteins will become amino acids, the glycogen will be broken down into glucose, and the triglycerides will be broken down into free fatty acids. Please note that you can take the glucogenic amino acids and convert them to glucose in a process known as gluconeogenesis. So, in the insulin land, it's feeding. The glucagon land is fasting. Insulin is a builder, #anabolic, but glucagon is a destroyer. #catabolic. Insulin is protein anabolic, glycogen anabolic, and fat anabolic. However, glucagon is the opposite. Protein catabolic, glycogen catabolic, and fat catabolic. Insulin stimulates protein synthesis, glycogen synthesis, triglyceride synthesis. Put differently, insulin induces proteogenesis, glycogenesis, and lipogenesis. Conversely, glucagon destroys, breaks down. So, glucagon stimulates proteolysis and gluconeogenesis and glycogenolysis and lipolysis. Today we're talking about triglyceride synthesis, which is lipid synthesis. It is lipogenesis. I'm converting something small into something big. I'm going to lump together 3 fatty acids and 1 glycerol to make one tag molecule, triacylglycerol, which is the prototypical triglyceride. If you wish to see more biochemistry videos like this one in the future, please drop me a sandwich emoji in the comments. Who is the centerpiece? Who is the hub? Who is the sun around which all of the biochemical pathways revolve? The answer is acetyl-CoA. If you know your acetyl-CoA, you know your biochemistry. If you think that acetyl-CoA is an aftershave for men, there is no hope for you. You can eat carbohydrates, you can eat proteins, you can eat lipids. Regardless of what you eat, you're gonna make acetyl-CoA because that's the central hub of biochemistry. Even if you drink alcohol, it will become acetyl-CoA. And then what? This acetyl-CoA will delve into the tricarboxylic acid cycle, also known as the Krebs cycle, also known as the carboxylic acid cycle. This will give you some ATP, which is energy, and will give you some NADH and FADH2, and will give those to the electron transport chain in the mitochondria to give me more energy. This pathway is known as lipid degradation or lipolysis. And we have talked about it before, and we talked about ketogenesis and ketogenolysis before in this biochemistry playlist. When you break down triglycerides, they become 3 fatty acids and 1 glycerol. When you build triglycerides, you will need 1 glycerol and 3 fatty acids. Why does your body prefer to store energy in the form of fat rather than carbohydrates or proteins? The answer is simple. Because fat will store 9 kilocalories per gram. But the terrible protein, only 4 kilocalories per gram. Terrible carbohydrates, 4 kilocalories per gram. Even the low-life alcohol, 7 kilocalories per gram. Not 9. So that's why your body prefers to store energy in the form of fat. Because it gives you more bang for your buck. It gives you more calories per gram of stored food. Lipids are super important, super dense, more bang for your buck, more kilocalories for your gram. Lipids are not soluble in water. They are not polar. They are non-polar. They don't like water, so they are hydrophobic. But lipids love lipids, lipophilic. Lipids are non-polar. What do all lipids have in common? They have long-chain fatty acids except cholesterol. That's why, technically speaking, cholesterol is not a real lipid. However, it acts like a lipid, behaves like a lipid, gets stored like a lipid, so we just lump it with the lipids. But technically, if you want to be precise, it's not a real lipid because it does not have long-chain fatty acids in it. What are the types of lipids that we have? We have triglycerides, we have cholesterol, we have essential fatty acids, we have phospholipids, and we have fat-soluble vitamins. What's the difference between triglycerides and phospholipids? Well, both of them are lipids. However, this is the difference. Triglycerides contain 3 fatty acids and 1 glycerol, but phospholipids need a phospho, so they will sacrifice one of the 3 fatty acids and put a phosphate instead. So you end up with 2 fatty acids instead of 3, 1 phosphate, and 1 glycerol, just like triglycerides. So the difference is here. Instead of 3 fatty acids for triglycerides, you have 2 fatty acids and 1 phosphate in cases of phospholipids. Cholesterol does not have fatty acids. Sphingolipids do not have glycerol. Why do we call cholesterol cholesterol? It ends in OL because it has an alcohol group. It's called stero because it has a sterol ring, and choly because it's made in the liver. You will recall that if this is glucose and this is pyruvate, then if you want to go from glucose to pyruvate, we call this glycolysis. But if you want to go the other way around, we call this gluconeogenesis. We have talked about both of them before. I will just remind you that in glycolysis, we go glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1 and 6-bisphosphate, and then DHAP, which is dihydroxyacetone phosphate. This can go back and continue glycolysis, or it can be diverted somewhere else, such as to make glycerol 3-phosphate, and this is super important. How do you convert dihydroxyacetone phosphate to glycerol 3-phosphate? By an enzyme known as glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase. Anytime you hear the word dehydrogenase in biochemistry, think oxidation reduction. Take that glycerol 3-phosphate and then you can combine it with 3 fatty acids to give you triglycerides. And this is called lipogenesis. It's also known as triglyceride synthesis, which is today's topic. If you wish to download these doozy colorful notes, go to medicosisperfectionalis.com. What does insulin want? Insulin is anabolic. Insulin is in the feeding state. So insulin wants you to build up fat, to build up triglyceride. It wants lipogenesis. However, where does insulin want to put my triglycerides? Insulin does not want them in the blood. Insulin wants your triglycerides to be stored in adipose tissue or fat cells. So insulin will actually destroy and break down the triglycerides in the blood so that you can take those free fatty acids, push them into the adipose tissue, and build them back up again to triglycerides. But what's the difference? Well, this is triglycerides in your blood, but that's triglycerides in your fat. And it's the triglycerides in your adipose tissue that will help you store energy for a later rainy day. How did insulin break down the triglycerides in the blood? By stimulating lipoprotein lipase enzyme. Any lipase is an enzyme that breaks down lipid, lipase. But insulin will stimulate the process of making triglycerides inside the adipose tissue. If you eat lots of sugar, you will be fat. I mean that literally. How come? Glucose can give you glycerol, and glucose can give you free fatty acids. One glycerol plus 3 fatty acids, and now we have a tag, a triacylglycerol, which is the prototypical triglyceride. Glucose, through glycolysis, can give us dihydroxyacetone phosphate. Glucose, through glycolysis, can give us pyruvate. Dihydroxyacetone phosphate can become alpha-glycerophosphate, which can become glycerol. Boom! Triglycerides. Pyruvate can become acetyl-CoA, malonyl-CoA, free fatty acid, and then triglycerides. So, the more glucose you eat, the more fat you will store, all other factors being equal. So tell me more about triglyceride synthesis. What is the quintessential triglyceride? It is called triacylglycerol. This is triacylglycerol. Notice it's called triacylglycerol, which means I have 3 acyl groups and 1 glycerol. So let's start with the glycerol and then add 3 acyl groups so that we end up with triacylglycerol. Okay, let's start. Remember that the more sugar you eat, the more fat you become. So glucose can give you dihydroxyacetone phosphate, which will later give you glycerol 3-phosphate by the famous enzyme known as glycerol 3-phosphate dehydrogenase. This usually requires ATP to come in and ADP to leave, which tells you that it takes energy to build up fat and store it for a rainy day, just like it takes energy and investment in order to save money for a later rainy day. As for glycerol, if you add a phosphate to it, it becomes glycerol 3-phosphate. This requires NADH to go in and NAD plus to leave. Remember that this NADH could have been utilized to make ATP, but again, it takes energy or an opportunity cost to make fat and store it for a rainy day. This is the glycerol, CH2OH, CHOH, CH2OH. If you add 3-phosphate to it, you have to sacrifice this hydrogen and replace it with phosphate. And now this is called glycerol 3-phosphate. By a doozy enzyme called acyl transferase, you will transfer or add the first acyl group because I want to end up with what? With triacylglycerol. If I need triacyl, 3 acyls, I better start by adding the first fatty acyl-CoA. Fatty acyl-CoA goes in, CoA will leave. So when coenzyme A leaves, you will end up with fatty acyl. Look at this glycerol 3-phosphate, and we're gonna add a fatty acyl to it. Remember that an acyl group consists of carbon double bonded to oxygen and single bonded to an alkyl group. Next, why not add the second fatty acyl? So fatty acyl-CoA is gonna come in, coenzyme A is gonna leave, and the enzyme is gonna be called fatty acyl transferase, or simply acyl transferase. And now, congratulations, you have added the second acyl group, carbon, double bonded to oxygen, and single bonded to the second alkyl group. Now, what do you call this? I'm going to call it DAG. D for di, but T for tri. So this is diacylglycerol phosphate, or simply diacylglycerol. Okay, why not add the third fatty acyl? Okay, let's add a fatty acyl-CoA. But CoA is going to leave the chat, so you end up with the third fatty acyl, which will be attached here. Here's the first acyl group, here's the second acyl group, here's the third acyl group. This is the glycerol skeleton. So, what do you call it when you have 3 acyl groups and 1 glycerol? How about triacylglycerol? That's just a beautiful name. What is the key rate determining step in triglyceride synthesis pathway? This doozy right here. It is the third enzyme that is called acyl transferase. And because it added acyl to DAG, we're going to call it DAG acyl transferase or diacylglycerol acyl transferase enzyme. This is the key or rate limiting step or rate determining enzyme. And because this is lipogenesis, it belongs to the insulin world, not to the glucagon world. And that's why glucagon hates this enzyme's guts. So glucagon is going to inhibit the acyl transferase. So let's regulate the triglyceride synthesis pathway. If you eat lots of sugar, you will become fat. So eating lots of carbohydrates, meaning lots of glucose, you're going to end up making more acyl and more glycerol. More fatty acids and more glycerol, you lump them together to make triglycerides. Insulin, feeding world. When you're feeding too much, you will store too much for the future. The more sugar you eat, the more obese you'll become. Conversely, what's the opposite of insulin? The glucagon world. Not just glucagon, but glucagon and his friends, such as epinephrine. Now, do you want me to make another video on phospholipids? Please comment below. If you want to learn more about insulin, the types of insulin, how to calculate the dose of insulin, to learn about cortisol, estrogen, progesterone, thyroxine, and more, download my endocrine pharmacology course at medicosisperfectionalis.com. To learn about cardiac arrhythmias, myocardial infarction, strokes, acute respiratory distress syndrome, diabetic ketoacidosis, and many topics of emergency and toxicology, download my emergency medicine high yields course at medicosisperfectionalis.com. It comes with videos, notes, and cases. There is another course on obstetrics and gynecology, and you can download it on my website as well. There are more than 600 premium videos available on this channel when you click the join button and choose the highest tier. Please subscribe, hit the bell, smash like, support my channel on Patreon, PayPal, or Venmo. Go to my website to download my courses, notes, and cases, or if you would like me to personally tutor you. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine, chemistry, and physics make perfect sense.